Excellent. Thank you, Nimi. I want to make sure we save a little bit of time for the audience Q&A. But before we do get to that, I do want to hear from both of you. And Nimi, perhaps I could start with you on this. And I think we would be remiss not to talk about the patient's voice in all of this. So Nimi, and then of course we'll go over to Bob. What are the considerations for use of patient experience data and incorporation of patient voice in cell and gene therapy product development and regulatory review and decision making? How much does that patient voice, how seriously is it taken?
Thank you for the question, Erin, and we truly would be remiss not touching that question, and not a new question and not unique to cell and gene therapy products. Under PDUFA 6 FDARA, and then later on, mandated in 21st Century Cures Act, FDA and industry continue to advance the incorporation of patient experience in medical product development and review. Specifically, per statute, FDA is directed to make public a brief statement regarding the patient experience data and related information, if any, was submitted and reviewed as part of such applications. And so, my first thought would be, we all, FDA and industry, should continue to collect, use patient experience data and submit to FDA for their reviews. And for FDA to make public their use of that patient experience data, whether these sponsors submitted it, whether it informed their decision making, and how, and that kind of information would really help advance the use of patient experience data to inform both product development and regulatory decision making. And specific to cell and gene therapy products, I mentioned the PFDD workshop that FDA held as mandated under PDUFA 7 last November, and they recently issued that report, and looking more carefully through these approaches to facilitate capture of patient experience data and involvement in clinical studies, including identification of challenges and gaps, considering available therapy for some indications, and continuing unmet medical need by the patients that could be met by this new product modality, cell and gene therapies.
Thank you, Nimi. That's wonderful. And Bob, I'd like to ask you the same question. What are the considerations for use of patient experience data and the incorporation of a patient voice as we develop and go through the regulatory review for decision making?
Over the years, I've seen the patient voice probably be the one instrumental point and factor that is leading to a change in thinking at the FDA. And what do I mean by that? Looking at the conditions for accelerated approval in these serious life-threatening conditions, particularly based on biomarkers. And the traditional thought was that once you get a product approved, you look at the biomarkers and you can validate it, and then you can use it for a follow-on product or someone else's product. Rarely, if ever, would it be used as a biomarker for a condition to be utilized for accelerated approval, but this is recently happening. And just take a look at in the Duchenne muscular dystrophy arena and the influence of the patient voice, or in this case, the parent voice, that's very impactful. So with that information and bringing that to the advisory committee and also to the FDA reviewers, this has a tremendous impact. What's important to the patient? What factors? And I can also allude to just a very recent incident where there was a rare bone disease in children. And in these young boys, they became paralyzed over a period of time, couldn't walk, couldn't ambulate, couldn't do their daily activities of living. And there was a concern because using the therapy would cause, in some cases, premature bone closure of the epiphysis, resulting in short stature. And so the feedback from the parents was saying, let us decide whether we want our young children in wheelchairs or being a little shorter in stature. Don't deny us that opportunity. And that resonated, and the product was approved recently. And so that's very important. And in fact, that voice is so important, it's been recommended, and I would also recommend it, to bring in certain circumstances, the patients and the patient voice to meetings with the FDA when you're talking about endpoints or your overall development program. That happens, and FDA does listen to those individuals. It really does have an impact. Brings that disease state to life and reality in what they're dealing with. So I think that's very important and has a really significant advance, and to continue to do that.
Absolutely, and wonderful sound advice to bring those discussions and those people and those patients to the FDA when these decisions are being made. So thank you for that. I want to now give a little bit of time to our audience Q&A. So thank you for submitting your questions. And first up, I have this first question, which, Nimi, if it's okay with you, perhaps you can start us off. Are there any in vitro techniques to predict potential safety concerns to save time on long follow-ups on post or pre-approvals?
Hi, Erin. Thank you for the question. I'm trying to digest the question and then have to opine on it. So, Bob and I also are together on the ARM regulatory advisory group that Bob chairs and I vice chair. And ARM has voiced their oral comments at the FDA workshop on post-approval safety and efficacy data collection back in April, where they alluded to ARM's thoughts on routine monitoring approaches in post-approval stage, which does include monitoring for malignancy concerns. And their consensus statement has been that routine approaches are appropriate for the disease area and the product, and should be considered based on the product-specific considerations. Also, our membership does not recommend surgical biopsies as a standard procedure for post-approval monitoring, given the risk, discomfort, or burden for patients associated with the procedure. And as such, we as an industry encourage and support the development of non-invasive methods for monitoring, including liquid biopsies and other markers. So that's overall that I'm sharing what has been the thinking in the field for this. Now, are there any in vitro techniques? Going back to non-invasive techniques, ultrasound is typically considered, a standard non-invasive technique for safety monitoring. In vitro techniques may be in development and not there yet. FDA has issued guidance on cell-free DNA. There's been discussions by, for example, Friends of Cancer Research on use of cell-free DNA as a biomarker for malignancy risk. So to answer Vijay's question, are there any techniques to predict? Yes, but they are in development. How much of a regulatory rigor they meet is yet to be seen. But typical non-invasive monitoring techniques, including ultrasound, are the ones that are recommended by an industry consensus. And if there is any signal seen in those, or if anything is picked up in those non-invasive monitoring approaches, then you follow up with a surgical biopsy or things like that to really understand the linkage. I hope that helps. Bob, did you want to add anything there?
Yes, this is an evolving field, particularly in bioinformatics. And if you look deep enough, you'll find particular genetic changes. And the question comes up, are they relevant? And so the deeper you look, the longer you look, you have these genetic changes, and are they relevant? And so there have been attempts made to talk about certain levels of detection that might be clinically meaningful. And then also those particular targets and areas that may be affected that could turn on oncogenes. And so, or turn off the suppressor to those oncogenes. So you turn on an oncogene and have a higher risk of malignancy. And so that's in its early stages, but people are delving in this area and setting up parameters on how to evaluate this information as it's being collected in its relevance to long-term monitoring for patient safety.
Thank you both for your detailed responses to that question. We're going to try to get to one more. And Bob, while you're up, I'd love to hear your response on this. We'll take a shot at this one. Experience on Type D guidance in biologics has been that the information is not binding in the same way as guidance from a Type BC meeting. Is that different for cell and gene therapies?
It's fairly new, and so haven't had personal experience with it or heard any feedback. I guess the questions come up, what is official information or comments or suggestion or guidance? And I always look at the feedback from the agency is very relevant and to be taken and evaluated and to address it the best way possible based upon your product and the science and what the FDA is requesting. If there's further clarification, there are opportunities to request clarification so you know exactly where the agency is coming from. I think in part of the things that we face is that the FDA per se sees all the cell and gene therapy products in particular classes with slight differences, and so they see the whole gamut of adverse effects and all activities, but based upon an IND, it's confidential information. So they can ask for things, and you may say, why did they ask for that? It's not relevant to our product, but they may be seeing something else. And this has happened, such as with the AAV vectors, where they saw certain toxicities. And so he actually brought what they were seeing to an advisory committee, and they identified 5 toxicities with AAV therapies for discussion in an open panel based upon research and what's available out in the scientific community.
Thanks, Bob. And Nimi, I'd like to offer you a chance to provide your response to that question as well.
Yeah, thank you, Erin. I'm looking at question 11 by Antonio that you posed, which is asking whether the information is not binding in the same way as it would be for a Type BC meeting, for a Type D meeting. We don't have FDA guidance yet. They are mandated under PDUFA 7 to revise their formal meetings guidance. We'll know more when that guidance is issued. But looking at 4 corners of the PDUFA 7 commitment letter, I have not picked up any difference in the binding versus non-binding nature of Type D or BC meetings. All Type A, B, C, D are formal meetings. Interact meetings are super early in development, pre-IND. Maybe that's the one that we may be commingling with. The difference between Type D, new meeting type, versus BNC is mainly in the fact that Type D is intended to be focused on a narrow set of issues. That's really primarily the difference. There are some differences in the timelines for when the meeting is granted and held and all of that, but really the main difference is Type D meetings should be limited to no more than 2 topics and no more than 3 disciplines or divisions at the FDA.